Hello, this is Bryce Christensen from Golden Helix. Today I'd like to show you some of the features of the Sequence Analysis module in Snip and Variation Suite in version 7. We'll begin by opening a project file. In this project, we have a data set from the 1000 Genomes Project Pilot 1. This is the Ceph European cohort. This spreadsheet has 60 rows corresponding to the 60 subjects in the data set. We also can scroll through and find that there are about 7.7 million columns corresponding to SNP variants that were identified in sequencing these 60 subjects. There's an associated map file we can view with important annotation information such as the chromosome physical position, the RSID of the SNPs where available, as well as the reference base and alternate base. Annotation information can also be viewed within our genome browser. Here we are viewing the minor allele frequency for all of the SNPs that were identified within a particular gene on chromosome 12. We also have several annotation tracks visible in the genome browser. There are a large number of annotation tracks available, and users are free to create their own annotation tracks as well. From the visible annotation tracks, we can see that we are within a particular gene, the RERGL gene, that there are several SNPs interrogated by the Illumina 1M Duo array within this particular gene, as well as on the Illumina 660 quad. When we zoom in closer, we are able to see finer details. For example, we can see that one SNP in our data set is interrogated by the Illumina 1M Duo and 660 quad. We can zoom in closer and see that it is a T to C change, as the reference allele here is a T. Even closer inspection allows us to look at the SIFT information that predicts that this particular mutation is to be well tolerated by any subject who carries it. The annotation tracks that we are viewing in the genome browser can also be used for analysis purposes within spreadsheets. Let's take a look at the original spreadsheet of SNP data. Here we have a spreadsheet containing over 7 million SNPs. It might be desirable to reduce this to a smaller number of SNPs using information contained in annotation tracks. For example, we might begin by reducing these SNPs to those that lie within exon regions. We can do this using filter by annotation, filter by exon region, and identifying the gene track that we would like to use to define our exons. Upon completion, we learn that only 50,707 of the SNPs were found inside exon regions. The SNPs outside of exons have now been inactivated in our spreadsheet, and we can create a subset containing only those 50,000 SNPs that are located within exons. Now we may want to reduce this set of SNPs even further. For example, we might want to remove SNPs that have previously been found and listed in the dbSNP catalog. To do this, we can go to Select, Filter by Annotation, filter by probe track membership, and identify the probe track for dbSNP 129. We can then inactivate the markers that are present in that probe track. Upon completion, we found out there were 10,000 of those markers which were not already listed in dbSNP 129. We can subset our spreadsheet now to those 10,000 SNPs. As a final filtering step, we might want to use the SIFT prediction algorithm to determine which of these SNPs are likely to be damaging. To do this, we select Filter by SIFT score, identify the SIFT track, and click OK. Upon completion, we find out that there are 1,204 SNPs remaining. We can reduce our spreadsheet one more time to this final group of 1,204 SNPs. These SNPs are all located within exons. They were not identified in the dbSNP 129 catalog, and SIFT prediction tells us that these SNPs are likely to be damaging variants. We now have several options available for analysis of these variants. For example, we could use chi-square tests or other standard statistics to compare them with a phenotype. We also have tests available such as the combined multivariate and collapsing tests that are designed for rare variants. A simple way to analyze them is the Count Variants per Gene tool. Using this tool, we can identify gene regions and count the number of variants identified within each gene for each subject. We now have a spreadsheet where the map contains gene names, and the fields contain a count of variants that were identified for each subject in each gene. We may wish to join this together with a phenotype spreadsheet. After joining, we have our phenotype together with these gene counts. We can run association tests then. This test shows us where our cases and controls have a difference in the burden of rare damaging variants. Test results can be plotted in a genome browser for further exploration.